Where'd that come from?
It's nanotech. You like it?
A little something.
Mr. Stark, I'm being beamed up.
Give me a little juice, Friday.
You said save the wizard.
We're too high up. You're running out of air.
Yeah, that makes sense. Oh, come on! Oh my God! I should have stayed on.
So you take one problem and you use it to solve another problem. We'll talk about the genesis of all of this. This is our nano cars. So we built these little cars. They have 4 wheels, independently rotating axles. This motor spins. If you shine a light on it, this little molecular motor will spin at 3 million rotations per second. And it will move this car across this surface when it spins. So this is looking at it much more slowly than that motor normally turns. It's 3 million rotations per second. And it will move a car across a surface. Now, these cars we just thermally heated this and the car started moving. So here's a car pointing and it's moving across this surface. And so you can see a car moving. And so these are nano cars. And then we just took the motors and hooked peptides on these to recognize certain cell surfaces. So there are these motors. The motors were known. They were called Faringa motors. We just built them into a car, right? We had a lot of fun with cars for many years. We'll talk about that. And then we used the motors to drill into cells. The motors by itself just drill into a cell and pop holes in it. So this is a bacterium, and you see the whole cell wall is just gone. And its guts are just spewing out. So you can drill into a cell with these things and kill them. So this is a rat. This rat has had its spinal cord completely cut in two. Now, we don't do the animal studies. That was done by somebody else. So this spinal cord was completely cut in two at the base of the neck. And then we put in 1 drop of a polyethylene glycol solution of graphene nanoribbons. Remember I told you that we made those graphene nanoribbons by splitting open carbon nanotubes? Put 1 drop in the gap there, and then after 2 weeks, the rat gets up and starts walking. Has anybody seen a YouTube short on that? Yeah. Okay, yeah, we made a YouTube short on this, and it's got like 5 million views. And then after 3 weeks, she's doing wonderfully. And so you see there, there she is after 3 weeks, scored a 19 out of 21 on mobility scale after 3 weeks. 21's optimal mobility. And she scored 19 after 3 weeks. That is with a totally cut into spinal cord. It's interesting, you can take these graphene nanoribbons now, and you put them in this gap. So you've cut the spinal cord, now it's in 2 pieces. And this is just a solution with the graphene nanoribbons. And you do this with the spinal cord. You just open the gap and touch it back together. In doing that, you've gotten these graphene nanoribbons to now align. It's called shear flow. Logs are in a lake, all disordered. As soon as they start going down the river, what happens? They all align with the river. That's shear flow, all right? So all the logs align with the river. Same with the graphene nanoribbons. They will align now just by doing this in that polymer gel that's there, and now they've aligned. And now when the neurons start growing, they grow from top to bottom and bottom to top. But normally, they just miss each other. So it's like ships in the night, and then they never reconnect. But here you have the graphene nanoribbons, and we had known from a bunch of other studies that the graphene, that cells, neuronal cells, like to grow on graphene. Well, why do they like to grow on graphene? Because graphene is conductive, and neurons like to grow on conductive substrates. It's because they're always sending out electrical pulses. And so what happens is now you have all these billions of them in there, in this gap. And so they'll start growing along the graphene nanoribbon. And some are growing from the bottom and some are growing from the top. And now they're growing along these highways. And now run into each other, and then they reconnect. And so the first 2 weeks, the rat was just sitting there just kicking around because the whole brain has to remap. Because what was the connection to the little toe is no longer the connection to the little toe. It's to the little finger now or whatever. All the connections have to remap. But just like our brains mapped it one time, it remapped it again. So this is why a little child, a newborn, you give them food, they don't know what to do with it. They don't even know how to hold it. And then when they learn to hold it, they stick it in their ear. They don't know how to direct it to their mouth. But after it's mapped, they've learned this thing and knows exactly where to put the food. The brain has mapped this. So the brain has enough plasticity that it can just remap the connections. That's what we're talking about. We remap these connections. So the National Nanotechnology Initiative, sort of like how nanotechnology came about. This is Richard Feynman. He won the 1965 Nobel Prize in Physics. He gave a lecture in 1959 and then wrote a paper about it in 1960 called There's Plenty of Room at the Bottom. So this is a PDF article that you can get online if you just did Plenty of Room at the Bottom, Richard Feynman. This is a guy who would think differently than everybody. He was an amazing guy. Some people don't like him because he was a little cocky. And you can watch bunches of videos of him on the internet. He died, 25 years ago, no more than that, maybe 30 years ago, something like that. But amazingly colorful guy. And he talked about how at really small scales, there's all these things that you can do, and we never really work at these scales. Now, fundamentally, when you're synthesizing molecules, you're working at those scales fundamentally, but he said there's so much other science that can be done at these small scales. So he has this lecture and this paper, There's Plenty of Room at the Bottom. And he wrote several books, but this book, Surely You're Joking, Mr. Feynman. And so the foreword is written by Bill Gates. This guy was amazing. If you ever just want to have a grand time reading a book, this would be one of those. It's just a lot of fun to see all the different things that he did. Just an amazing guy. So he was a young guy during the Manhattan Project when they were making the nuclear bomb. Did anybody see that recent movie? What's it called? Oppenheimer. Did anybody see Oppenheimer? Do you remember a guy playing the bongos? Yeah, there was a couple times there was a guy in the back playing the bongos. That's him. He loved playing the bongos. He was just a young guy during the Manhattan Project. And so he's also the guy, remember there was I think it was the Challenger disaster. There was one 40 years ago. One of the space shuttle blew up on launch. They had like a school teacher in there, and there was an astronaut from Israel on it, and it blew up shortly after launch. And he was the guy who figured out what happened, like almost instantly, because it was a very cold day that day. And so he looked at the rubber seals, and he just stuck it in cold water. And then he took the rubber seal that did this and just cracked apart. The rubber seals were never made to operate at sub-zero temperature. And it was just anyway, sharp guy. Okay, so he had this Plenty of Room at the Bottom, but then Mikhail Rocco, goes by Michael Rocco, he was at the NSF and he said, let's build a program around this nanotechnology, because Rick Smalley, who was here at Rice, Rick Smalley passed away in 2005. He won the 1996 Nobel Prize for his discovery of Buckminster fullerene, C60. He, along with Bob Curl, who just passed away last year, and also Harold Kroto, who passed away about 3 years ago, who was from the UK here at Rice visiting when the discovery was made. Rick and I were good friends, and so Rick was really pushing this thing of nanotechnology because he had just discovered C60. Michael Rocco said, let's have a program, a big national program around developing nanotechnology. Also, on the Hill was Neil Lane. Has anyone ever met Neil Lane? This is Neil Lane and Bill Clinton. Neil Lane is a professor here. He's now in the Jones School and in public policy, but he was a physicist at Rice, and then he became provost and then maybe president in Colorado, and then he was selected to be the White House, then he became head of the NSF, head of the National Science Foundation. Then Bill Clinton chose him to be White House science advisor. he pushed this whole idea of having a national nanotechnology initiative with Bill Clinton. The idea was to have a backhoe where you could shine a light on this, get this to go to the cis, and then in that cis form, these two nitrogen atoms can chelate a metal, so they can chelate a metal, and then you could move it back, you could move it to where you wanted it, and then shine a different wavelength of light. It'll go back to the trans and drop off the metal. So you see that pick up metal and drop it off. So now you can do, buildings. So if you get enough of these building for you. So the whole idea was to think about bottom-up construction this way. We made all sorts of different wheels. And so you see the synthesis that you go through. And students love this stuff who like organic synthesis. They like to build molecules. It's just what they like to do. So they come in and they do this kind of coupling chemistry here. They do this coupling. They get the 4 alkynes, which would be the 4 axles. And then they put on this organometallic complex. So now you get this big organometallic complex. So these are really big wheels here that are on the edges of this. This is the space filling model. This is if you filled up the van der Waals radii. And so you can see the ruthenium atom is actually buried inside of this. It's very hard to see because you have all of these phenyl groups and these ethylene groups here. But anyway, so it has the big wheels on it. Then we wanted to try making self-assembling metal complex nanocars. So could you have 2 halves of a car, and then you drop in a metal between them and they just start assembling. So you build you build half a car and it assembles into a whole car. So here's the half cars in a solution without the metal added. And when you add the metal, the whole color changes and you get these whole cars here. So that would be the idea. So we went ahead and built these structures doing the organic synthesis. So there's the half car, and here's 3 nitrogen atoms, and then if you put 2 of these together with iron would sit right in the middle and pull these 2 halves together, and then you get the whole car. So you see that would be sort of the arrangement here. And so that worked. So, when you see a yield here, that was the yield of the actual reaction. And so you can see here's the half car. As soon as you complex it, you get the whole car. And the color changes of the solution too. The half car, it doesn't absorb light in the visible region. If you just take ferric chloride, which is just yellow solution, you add the ferric chloride to the half car, and then boom, you got the complexed car. And then you're seeing the wavelength much further out. And the NMR substantiates this. Then there's a hydrogen bonding car. Just like DNA hydrogen bonds, you get the GC, for example, and they're hydrogen bonding between each other. Could you do the same type of thing, just take a half car and have it assemble into a whole car? You say, well, why are we doing this? And I'm like, why not? What do you want to do for a PhD? You're building new structures, new nanostructures. If we can learn to make cars rapidly, we could think about doing construction with them. So could these then generate full cars this way? So we just took this hydroxypyridine and that we iodinated, and we put the alkyne on it, clip off the TMS group, and then do the Sonogashira coupling. So now here's a half a car, but it doesn't stay as the half car. This dimerizes directly to the whole car. And we know it's dimerized because you can see in the mass spectrum, you can see the molecular weight that's consistent with the dimer, not the molecular weight that's consistent with the monomer. So this thing is dimerized directly to the full car. How about trains? Could we build trains this way? That's the idea. And so the idea is that if you can have some terminal wagons, so this might be like a caboose, and if you could make a covalent dimer so that if you take these bromides and you're on a copper surface, copper will cause these to undergo what's called an Ullmann coupling. And that would make this covalent dimer. That was the idea. And or if you wanted just this to extend, you put the bromide on both ends and you have it extend. The interesting thing about this is that if we rotate, if you rotate about this single bond, that's going to place this ring up here and this ring is going to end up down in this direction. So you see, instead of them being parallel, there'll be a cross correlation or anti-parallel to one another. And you can end up seeing that on the surface. So remember, there's this parallel conformation, and then there's this cross conformation. So when this is on a surface, we should end up seeing both. And that's exactly what happens. You put this on a surface and you end up seeing both. So you can see here's a parallel conformation, and here's a cross conformation. All right, and you can tell by the distances here that this is right at the distance that this should be across. But what happened is that what we expected these to dimerize like this, that the copper would take out the two bromides and it would dimerize. But what's happening is the copper gets in between, it spits out the bromides, but the copper atom is staying in between. You say, well, why does that occur? And the reason that occurs is when you're in solution and you're trying to put two aryls together, two bromobenzenes together, what happens is they'll get on a copper atom and they'll be in a cis conformation. And from that cis conformation, the copper atom is ejected and the two arenes end up sticking together. But this can't get into a cis conformation. They're going to stay trans to one another. And the reason they stay trans is purely because they don't have enough freedom on the surface. It's not like they're in solution. So the copper can't move them to get the cis conformation to eject. So what we ended up, because you can tell by the length, so if the copper had been spit out from axial to axial should be 18 angstroms. But what it's turning out to be is to be 20.8 angstroms. And that means the copper atom is sitting in there. If the two bromides had remained in there, it'd be 25 angstroms across. If one bromide had been in there, it'd be 22 angstroms across. But it turns out this distance is right at 20.8 angstroms, this distance. And you can actually see between these, you can see copper atoms sitting between these. So you just take this half car, you just take this structure, put it on the surface, and what happens on the surface, it immediately polymerizes. So you can see this polymerization of chains. And once in a while, and so it's mostly parallel conformation, that should be the lowest energy conformation, but once in a while you see this cross conformation, where the two axles are anti-parallel. So you can see this cross conformation here. And when you look closely, you can see there is the copper atom sitting on the surface, and the imaging was done by a colleague of mine, Leonard Grill, and his group. And the synthesis was done by Takashi Sasaki in my group. And so you get these polymeric structures, and this is what we expected. We expected these trains to form, and they formed like this. So think about this. You can't build from the top down this small. You could never make large structures and whittle them down to a small structure like this. These are all made from the bottom up through organic synthesis, and then you just put these on the surface, and they're pulling copper atoms off the surface to do this chemistry.